The protein data bank includes 3D structures of all biological macromolecules, including proteins, nucleic acids, carbohydrates, lipids, and their various complexes. Carbohydrates play many different roles in biology. Molecules like glucose, fructose, and starch function as the source of energy in cells, while agarose in seaweeds, cellulose in plants, chitin in many insects play key structural roles. Carbohydrates are also frequently found linked to the extracellular phase of membrane proteins and some secretory proteins. These glycoproteins play important roles in recognition and binding. Carbohydrates are hydrates of carbon, so their building blocks or monosaccharides are primarily composed of carbon, hydrogen, and oxygen atoms. Some monosaccharides also include nitrogen, phosphorus, and sulfur atoms. Although these molecules exist in their linear form, more frequently they are found in the form of 5 or 6-member rings. Carbohydrates differ from other biopolymers like proteins and nucleic acids in two major ways. The first unique feature is the versatility in their binding. All amino acids form peptide bonds to connect with other amino acids and form proteins. Similarly, all nucleotides are connected to each other via phosphodiester bonds to form nucleic acids. The glycosidic bonds are a little different. Monosaccharides can form covalent bonds at a number of different points around it. This allows saccharides to form both linear and branched chain structures, unlike proteins, DNA, and RNA. In addition, glycosidic linkages can even be formed between saccharides and other macromolecules, forming glycoproteins and glycolipids. Unlike DNA, RNA, and proteins, carbohydrate polymers are not encoded by the genome. The specific order for linking different monosaccharides is determined by a series of enzyme reactions that act in concert. For example, the process of N-linked glycosylation, adding saccharides to specific asparagine residues to form a glycoprotein, begins in the endoplasmic reticulum. This is then enzymatically trimmed to a core saccharide to further be built up by the action of other enzymes to form the final glycosylated product. There is no reference sequence of saccharides or other genomic instructions for these steps. Therefore, there is no mechanism to make edits or corrections when errors are made. In fact, in living cells, decoration of proteins with carbohydrates may vary slightly from molecule to molecule. Many structures in the PDB have carbohydrates either covalently and or non-covalently bound to proteins and lipids. In most cases, this is only a small part of the carbohydrate structure, the part that is ordered and visible. Parts of the carbohydrate molecule that are mobile or cleaved during sample preparation are not visible in the experimental data. Keep this in mind when exploring carbohydrates in the PDB.